All right, welcome to chapter 16, where we are going to be taking a good hard look at DNA and some of the processes that DNA undergo in order to make life possible. One of the main things we're going to cover in this little video cast, will be to look at the history of how we came about to the conclusion that DNA is the main substance of inheritance and one of the most celebrated molecules of our time. I mean, the molecule is called deoxyribonucleic acid, DNA for short, and it was discovered, or I shouldn't say discovered, but the two people that are primarily responsible, Watson and Crick, who in 1953 viewed this very elegant, iconic image of the double helix, two molecules that wrap around each other, that you see in any biotechnology company probably is going to have the DNA symbol incorporated into its logo somehow. But there actually was a third person. Her name was Rosalind Franklin, and she did some really important experiments where she was bouncing X-rays off of DNA that had been crystallized. When the X-rays hit the crystallized DNA, it would scatter off and create a pattern. As it was, Watson, who also did some work with X-ray diffraction, recognized that pattern right away as the double helix pattern that the DNA molecule would have to have in order to see scattering like this. Franklin won the Nobel Prize as well, but she is not nearly as famous as Watson and Crick because she died before the Nobel Prize was handed out for this model. In the early 20th century, they were not certain as to what the molecule of inheritance was. We're doing a lot of stuff in the 1800s and early 1900s with genetics, but no one really understands how this hereditary information is being passed on from generation to next, parent to offspring. Mendel called them factors. Of course, today we know that they are genes located on the DNA molecule. So let's talk a little bit about some of the history and how we got to where we are today. In our T.H. Morgan, his group showed that genes were located on the chromosomes. If you remember back to the previous chapter talking about fruit flies, and they knew because eye color was a sex-linked trait, it was showing up on males way more than it was showing up on females, that gene had to be linked to chromosomes. So we've already talked about that, but chromosomes are made up of two things. It's about 50-50. It's about 50% DNA and about 50% protein. So there was a big fight then that began, a philosophical fight among biologists during this time period as to what is the molecule that's responsible for this genetic material. Is it the protein in the chromosomes or is it the DNA? When we look at the role of DNA in heredity, the study of bacteria and viruses are what really led us to the conclusion that it is DNA that's the hereditary material. So, all right, so the first experiment that was done in 1928 by a guy by the name of Frederick Griffith. Griffith was primarily involved in working with a couple different strains of bacteria. One caused pneumonia. It was a pneumococcus, and there were two different types of pneumococcus. There's what we call the smooth strain, and there was the rough strain. The smooth strain had a very smooth outer border around the cells. The coccus means that they're round cells. And then the rough strain had a very jagged edge around those spherical cells. And the smooth strain was pathogenic. In other words, it gave you pneumonia and you potentially would die from that. The other one was harmless. And so he did an experiment that showed that the smooth strain could take the rough strain and cause it to undergo a transformation where it went from being harmless to being pathogenic by taking on DNA. So let's talk a little bit about that experiment. He did a couple really important things. Number one, these are smooth strain cells. We'll call them S cells because they are the smooth bacteria. And he injected this harmful pathogenic bacteria into some mice, and of course the mice got pneumonia, they died. He took some R strain, rough strain cells, and again they have the rough outer edge because they're not inside a capsule. When we get to the chapter where we're studying bacteria, bacteria that are encased in a capsule like this are much more difficult for the body to destroy, therefore they can do their thing, and that they're the more dangerous type of bacteria. Here we don't have that capsule, so the immune system can get at these cells and destroy them, therefore the mouse lives. Okay, so what he did next, I mean, he expected both these results, nothing shocking here. Alright, so his third experiment, he took the dangerous bacteria, the S cells, and he heated them to the point where he killed the bacteria, and then he took a sample of this dead disease-causing bacteria, injected into some mice, and of course they lived. Of course they lived, the bacteria are dead. Here's where it got to be interesting though. For some reason Griffith decided, what if I mix my living R cells and my dead S cells? In both cases the mice lived. And so you would expect that in this trial, putting these two things together shouldn't make a difference. The mouse should live. It should not be a problem at all. But when he injected a serum that had both the dead disease-causing bacteria and the live non-disease-causing bacteria, the mice started to die. Okay, now what the heck is going on here? These mice should be living. So he performed an autopsy, looked at them underneath the microscope, and what he found was living S cells. And so Griffith's hypothesis was that there must be something inside these dead cells that were taken in by the living cells and a transformation occurred. In other words, these R bacteria, R cells, were transformed into S cells. All right, so about 15 years later, famous experiment number 2 involving DNA happens. A guy by the name of Avery leads a group of scientists that are working again with these same pneumococcus bacteria. And they're trying to figure out what was the mechanism that caused the transformation. How did the R type become S type? So what they did is they made an extract from the heat-killed bacteria, and they treated that extract with enzymes. In other words, they used the enzymes to destroy all the proteins, destroy all the lipids, the carbohydrates, even nucleic acids like RNA, not DNA though. So when they put the extract into a culture of R cells, transferred, they became S cells. So then they repeated the experiment, this time using enzymes that would break down DNA, but allowing the proteins and carbohydrates, etc., to remain intact. This time the DNA was destroyed and transformation did not occur. So they had their control group and they had their experimental group, and this conclusively proved that DNA was the transforming factor. The problem is, again, it's 1944. We really don't have good technology in terms of microscopes and whatnot, and so most of the biologists remained very skeptical. They felt that something had to be wrong with Avery's experiment, something had to be going on with Griffith's experiment that was causing the transformation, and it couldn't be DNA. DNA just was not a complicated enough molecule to house all the genetic information that we see in living organisms. It had to be the only thing that caused it. So they knew it had to be DNA. But again, at this time, people were not willing to buy the argument that DNA was the material responsible for the transformation. So we needed something more conclusive. Well, about this time, we're getting into the 1950s now, our technology is starting to improve. We're getting microscopes that are actually pretty good. You need a really, really good microscope in order to see viruses. It's one thing to look at bacteria, but in order to see a virus, you're going to need a pretty darn good microscope. So we really couldn't do some of the important experiments that were done until we got the technology up to speed. Well, there was a group of scientists that were working with bacteriophages. What a bacteriophage is, by the way, it's the T2 bacteriophage. This specific bacteriophage is a virus that attacks E. coli, the E. coli bacteria. And what it does is it injects its nuclear material, okay, which is contained inside this little capsid here. By the way, the nice thing about viruses is they're only made up of two molecules. We have this protein that makes up the capsule, the outside of the virus that you see, and then the nuclear material, DNA. Some viruses contain RNA, but a lot of these bacteriophages contain DNA inside the capsule. So anyway, what this T2 bacteriophage does is it attaches itself to the cell, it injects its DNA inside. That DNA material takes over the cell. It basically destroys all of the bacteria's genetic material so that only the viral DNA is left. That viral DNA then starts to instruct the cell, hey, let's make some T2 bacteriophages. And it will continue to do that. This cell will become a factory for this particular virus, and it will keep going, keep going, keep going until eventually the cell ruptures, releasing all these new viruses. The picture is an electron microscopic picture of the T2 bacteriophage doing its thing. You can see that where it's dark, shadowed, there's nuclear material inside. You can see the nuclear material actually being punched through that cell membrane, cell wall, and now it's empty on the inside. So whatever was inside that bacteriophage is now inside the cell, and it's doing its thing. All right, so in 1952, a couple scientists by the name of Hershey and Chase did a landmark experiment working with the T2 bacteriophage. They designed an experiment that would show that only one of the two components of the T2 would enter an E. coli cell during an infection, and therefore whatever that material is it DNA, is it protein, one of these is entering the cell, taking over the cell, and causing the transformation. So they think they have an experiment that finally will demonstrate it is DNA and protein. What they did was they made radioactive batches of the T2 bacteriophage. What they did was they knew sulfur is an element that is found in protein but not in DNA. Okay, so they grew this one batch of phages in a medium that had a whole bunch of radioactive S35. So these atoms of sulfur would become incorporated into the virus, incorporated into the protein that makes the virus, and therefore if that shows up inside the cell, we know that it is the protein that's causing the transformation. On the other hand, they had another batch of viral particles, molecules, whatever you want to call them, phages, and these viral phages were grown in a medium that contained radioactive phosphorus. Phosphorus is found in DNA, but it is not found in proteins. They put a little bit of radioactive DNA into a culture of E. coli, let it do its thing. They put another batch of radioactive phages into a culture of E. coli, only this time it was a different radioisotope, let it do its thing. And then they dumped all this stuff in a blender and ground it up and put it in a centrifuge and spun it until they had everything separated out. If the pellet contained cells, E. coli cells, that were full of this radioisotope, then they knew that the protein was the transforming mechanism. They didn't find any radioactive sulfur inside the cells. If the pellet from the next experiment contained radioactive phosphorus inside the cells, then they knew that it had to have been DNA that was being injected into the cell and causing the transformation. And of course, that is the results that they found. They did not find any radioactive sulfur. They only found radioactive phosphorus.